This video covers information on properties of the microscope, preparing wet mount slides, and using a dissecting scope. The image you see through a microscope isn't an exact projection of what's on the slide. Aside from the object appearing larger, it's also inverted. Curved lenses in the microscope cause the image to appear upside down and backward. This isn't usually obvious unless you're looking at something with an obvious left, right, top, and bottom, or attempting to move something under the microscope with your hands. Biologists use slides of the letter E to demonstrate inversion. Some of these slides are prepared with the E mounted to compensate for inversion, so be aware of the slide orientation before placing it on the microscope. Make sure to place it on the stage with the E positioned as you would read it, and then notice how it changes when you view it through the microscope. You'll also notice the inversion effect in a more obvious way during your next activity, when you attempt to measure the field of view. As I mentioned in the previous video, the size of the field of view varies with the objective you're using. This makes sense when you think about it, since you're zooming into a smaller and smaller area of your specimen. We measure the size of the field of view with its diameter. This is the length from edge to edge across the widest point. For the scanning objective, where the diameter is relatively large, you can measure it directly using a metric ruler. As you increase magnification and the field of view becomes smaller, you must determine the diameter mathematically. To measure the field of view on the scanning objective, place a clear metric ruler across the center of the condenser lens, as if it were a slide. Then, look through the oculars and line up the center of a millimeter mark on one edge of the field of view. This is where the inverted image becomes a problem. Moving the ruler short-circuits your brain a little bit. Left is right and up is down. Take your time and line up the ruler carefully. Once you have it lined up, count the spaces between the millimeter marks to determine the diameter of the field of view. If what you see were like what's shown on the screen, then this would be 1, 2, 3, 4, 5.5 millimeters. For objectives with higher magnification, the diameter of the field of view can be determined mathematically using the diameter measured under scanning. To calculate the diameter of the field of view for any objective, all you need to do is multiply the total magnification of the scanning objective by the measured diameter of its field of view, and then divide by the total magnification of the objective whose field of view you want to know. Let's look at a few examples. Remember, in our example earlier, we said the diameter of the field of view under the scanning objective is 5.5 millimeters. Now, let's calculate the diameter of the field of view for the 10x objective. To find the diameter on 10x, we need to use known information. First, we need the total magnification of the 4x objective. Remember, to determine total magnification, we multiply by 10. So the total magnification of the 4x objective is 40. Now, we need to multiply that by our measured diameter for the 4x objective. We said that was 5.5 millimeters. Next, we need to divide that product by the total magnification of the objective we want to find the diameter for. Since we want the diameter for the 10x objective, we need to divide by its total magnification, 100. 40 times 5.5 is 220. And 220 divided by 100 is 2.2. So the diameter of the field of view for the 10x objective would be 2.2 millimeters. This makes sense if you think about how the total magnifications compare. The total magnification of the 10x is slightly more than twice the total magnification of 4x, and the diameter of the field of view is slightly less than half as wide. As magnification increases, the diameter of the field of view decreases proportionately. Let's try another one. This time, let's figure out the diameter for the 40x objective. Again, we multiply the total magnification of the 4x objective, 40, by the diameter of its field of view, 5.5. Again, this is 220. This time we need to divide by the total magnification of the 40x objective because that's the objective we want the diameter for. So this time we divide by 400. 220 divided by 400 is 0.55 millimeters. Notice that the total magnification of the 40x objective, 400, is 10 times larger than the total magnification of the scanning objective, 40, and the diameter of its field of view is 10 times smaller. Now, let's try one more time with the 100x objective. Again, we start with the total magnification on scanning, 40, multiply by its measured field of view diameter, 5.5 millimeters, to get 220. This time we just divide by the total magnification of the 100x objective, so 1000. 220 divided by 1000 is 0.22. So the diameter of the field of view for the 100x objective is 0.22 millimeters. Now, let's learn a little bit more detail about microscope technique. I mentioned before that contrast is important to viewing microscopic images. Even with high magnification and good resolution, it can be difficult to see the structures of clear cells against a bright white background. Notice the cheek cells on the top. They almost blend into the background. The cells on the bottom have been stained with a dye called methylene blue to enhance their structures and improve contrast. You can also improve contrast by closing the iris diaphragm on your microscope. Closing the iris diaphragm also affects the depth of field. Depth of field is the vertical depth of a slide. You can explore the depth of field by slowly manipulating the fine focus on your microscope. Closing the iris diaphragm appears to flatten your image, allowing you to focus on more depth at once. Opening it makes your image appear more three-dimensional. You'll want to have the iris diaphragm open when exploring the depth of field with your fine focus. Exploring the depth of field allows you to determine the arrangement of objects in vertical space. Using the fine focus, you can determine the order that the objects come into focus. You'll be looking at a slide similar to the one shown here. You'll need to examine the area where the colored threads cross and determine their vertical order by exploring the depth of field. In the image shown here, you can see that the red thread is more clearly in focus, followed by the blue and then the yellow. This means that the red thread is on top, with the blue thread underneath it and the yellow on the bottom. In addition to viewing prepared slides like this, you'll also be making your own slide in the lab. Temporary slides prepared in the lab are called wet mounts. They're made by mounting a specimen in liquid between a slide and a thin glass cover slip. To prepare a wet mount, first place the specimen on the slide and then cover it with a drop of water. To prevent excess air bubbles, don't just drop the cover slip directly on top of the liquid. Instead, hold the cover slip at a 45-degree angle and pull it until it meets the edge of the water. When it meets the edge of the water, you'll see the water spread along the edge. Now you can drop the cover slip, forcing out most of the air bubbles beneath. Dry up any excess water and then place the wet mount on your microscope for viewing, just as you would any other slide. There are many variations on preparing a wet mount, such as staining the specimen under the cover slip. In your lab, you'll be making the simplest of preparations, pond water. For this, there's no specimen to place. The specimens you'll be viewing are already suspended in the water. You'll just need to put a drop of pond water on the slide and place a cover slip like I described. Pond ecosystems are teeming with aquatic microorganisms that can be observed with your microscope. Single-celled organisms, some photosynthetic, along with multicellular invertebrates. Explore around and find something interesting to draw in your lab manual. Maybe you'll even see some of the organisms shown here. When you're finished viewing your slide, make sure to dispose the cover slip in the glass box. Wash your slide and set it by the sink to dry. Some objects are too large or three-dimensional to be placed on a slide and viewed with a compound microscope. Dissecting scopes are useful for viewing these items more closely. The parts and functions of this microscope are a little different, so let's make sure you're familiar with them. Your dissecting scope has two 10x oculars, just like the compound microscope. Remember, these magnify the specimen 10 times. The objective is different, though. Instead of several objectives with their own specific magnification, there's a single objective fitted with a zoom lens. This allows you to smoothly increase magnification simply by turning the zoom knob. Increasing the magnification will cause you to lose focus on your specimen, so you'll need to bring it back into focus with the focus knob. Just like on a compound microscope, the stage for placing the specimen is below the objective. It's just a little bit closer to the base. On your dissecting scope, there are two options for lighting the stage. To view the surface of an opaque object, you use the incident light. It's adjustable, so you can move it back and forth to adjust the lighting. You can also illuminate translucent objects from below, using the light source underneath the stage. It shines transmitted light through a glass plate on the stage. I mentioned before that the dissecting scope is not capable of magnifications as high as a compound microscope. You should also be aware that calculating the total magnification is a little different too, because of the zoom objective. Here you can see that the zoom knob is marked with possible magnifications of the zoom objective. If you look closely at the labels, you'll notice that the magnification ranges from as low as 0.75 to as high as 5.0. It's difficult to see in this image, but there's a raised mark next to the zoom knob that indicates the current magnification of the zoom lens. In this case, the zoom is set to 5x magnification. To determine total magnification on the dissecting scope, you need to multiply the zoom setting, 5x, by the ocular magnification, 10x. So a specimen viewed at this zoom setting would be magnified 50 times. This is the maximum total magnification for this dissecting scope. Let's try one more example quickly. What if the zoom knob were turned to its lowest setting, 0.75? Then we would need to multiply 0.75 by 10 to determine total magnification. Remember, when multiplying decimals by 10, we just move the decimal one place to the right. This means the total magnification on this setting is 7.5x. The learning objectives you're responsible for are listed in your lab manual at the beginning of Laboratory 4. The ones specific to this video are listed here. Make sure you understand all terms listed in bold and italics. Be able to identify and explain the function and parts of the compound microscope and understand how to properly handle and focus it. These objectives will be tested in your upcoming quiz and lab exam. Contact your lab instructor if you need any help or anything's unclear. Good luck!